Can We Build Our Perfect Child by Ryan Webster. As time goes on within the genetic field, one of the primary questions still remains, even now more than ever, bringing forth the question to the forefront that is, can we build our perfectly envisioned child? What would this look like, and what ethical issues come along with that? Let's get right into it. First off, what is gene editing? According to the World Health Organization, genome editing is making changes to the DNA of a cell in an organism through removal, addition, or alteration of the genome. Gene editing has been made possible through the discovery of such systems as the CRISPR-Cas9 technique, which allows for this developed repair system derived from bacteria. Gene editing has already been used in somatic cells, which are the non-reproductive cells of our body in regards to aspects such as helping address HIV and sickle cell disease through in vivo techniques. However, when we discuss building the perfect baby, today we will be focused on the germline cells, which are the reproductive cells within the human that allows for changes to be made to offspring, affecting generations to come. So, how does the CRISPR-Cas9 system work? The system requires two things, a guide RNA and the Cas9 nuclease enzyme. It then follows three stages, recognition, cleavage, and repair. The process starts by the guide RNA targeting the Cas9 enzyme to the desired DNA site by base pairing with it. The presence of a protospacer adjacent motif, or a PAM, adjacent to the target site allows for Cas9 binding and cleavage. Then, after bound to the target DNA, the Cas9 enzyme will make a double-stranded break in the DNA, and this cut triggers the cell's DNA repair machinery. After the break, the cell repairs the DNA via one of two major pathways, non-homologous ends joining, NHEJ, or homology-directed repair, HDR. NHEJ is used to knock out genes and disrupt their function, while HDR uses a more precise mechanism that allows for insertion, deletion, or replacement of a sequence to knock in functions. Most cells prefer the NHEJ pathway, however, so it can be difficult to knock in genes. Because of its effectiveness and efficiency, CRISPR-Cas9 is the most utilized genome editing tool in all living cells. Now, let's take a look at how CRISPR-Cas9 is used for gene editing to treat things such as sickle cell disease. First off, sickle cell disease is caused by a mutation in the beta-globin gene, which produces hemoglobin. This mutation results in the production of an abnormal form of hemoglobin known as hemoglobin S, which contains a sickle shape, leading to all these complications. In this example, CRISPR techniques were used ex vivo to re-express HbF in adult red blood cells to reduce the impact of the sickle mutation. This was achieved through editing the promoter in fetal hemoglobin's gene in the patient's own stem cells, thereby reactivating fetal hemoglobin expression in a way to alleviate sickle cell disease. In doing this, they showed that the edited cells express more HbF, reducing sickling. Now that we've established the possible genome editing technique and an application, let's look at how that may be utilized in the future to modify babies. So how does this impact physical traits then? Geneticists have coined the so-called term designer babies in regards to the potential future babies which can be modified through in vitro fertilization using gene editing to create a baby with the most desirable traits. But how far away is that from reality? Based on our research, we can modify the outcome of a baby up to a limited extent. Technologies such as pre-implantation genetic diagnosis, PGD, allow parents to select embryos based on certain genetic markers, like choosing sex and screening for or against genetic disease markers. Based on the results, parents can choose the embryo they see most viable or fit. Some clinics even offer such traits as hair and eye color as well. In regards to complex traits such as intelligence or height, it is harder to modify that in regard as those factors are influenced by a vast numbers of genes and factors and therefore harder to manipulate. Also, many scientists are hesitant in using CRISPR-Cas9 techniques in regards to further modifications for human trials, as there are many ethical factors and unforeseen consequences that could arise from it. With that being said, the future holds many possibilities and breakthroughs using CRISPR-Cas9 that are inevitable. It will just come down to a debate on ethics and morality. With that being said, let's also take a look at the ethical implications once genetic modification on designer babies. One of the biggest factors holding advancements back in regards to genetically modified babies is ethics. In China, in 2018, a scientist went to jail after changing the genes that allowed HIV to enter the cell in utero to a pair of twins who eventually were born healthily. Along with that, many countries, including the United States, have banned embryo gene editing due to ethical concerns. Federal law also prohibits the use of federal funds on human germline gene therapy, including research on genetically edited embryos. While private funding may still take place, they still cannot proceed to clinical trials involving humans. Another moral concern arisen is the wealth gap between those able to pay for the procedures and those who can't. Most likely, only very wealthy individuals would be able to afford these procedures, which creates the emphasis on elitist status and creates unfair advantages in regards to a cycle of producing very smart and perfected individuals. The potential perpetuated cycle raises concerns as an ethical issue. Finally, another ethical issue that arises when using CRISPR in genetic modification is the argument that scientists are playing God. Because of this argument, there are many people against the idea of genetic modification as it is not viewed as a natural process. It is because of these ethical reasons why the scientific progress of genetic modification is not as far as it could be. With that being said, thank you for listening today, and that concludes my presentation. Here are my references, and I hope you enjoyed and have taken something important away from this topic today.